In this video, we're going to learn what genetic mutations are and how they can affect transcription and translation. So there can be a variety of different outcomes. You could have a silent mutation, which would have no effect on the cell. You could have a nonsense mutation, which puts a stop codon in the gene and stops translation, which would be really bad. You could also have a missense mutation where just one amino acid is inserted incorrectly. And then finally, you can have a frameshift mutation, which would change the entire reading frame of the gene, which would cause an entirely different protein to be translated. So first of all, a silent mutation has no impact on the cell or the protein. And the reason this is the silent mutation could be in an intron region. So remember, those are the non-coding regions. They get cut out after transcription before the gene is translated. Or it could be a mutation where it will still cause the same amino acid to be inserted into the protein. So if you look here, if we have CGU and there's a mutation to make it be CGC, both of those codons code for arginine. So if this mutation happened, it would be considered a silent mutation because arginine will still be inserted, so there will be no difference. The second type of mutation would be a missense mutation, and in this case, the wrong amino acid is being inserted into the polypeptide or the protein. And the example I have here is originally we had UGG, and that codes for tryptophan or TRIP. And if there's a mutation that causes UGC, this codon codes for cysteine or cis. So you would have the wrong amino acid placed in the polypeptide. Our next type of mutation will actually cause a shortening of the protein. So what happens is a stop codon is inserted instead of the intended amino acid somewhere along the way. So only a part of the protein will be built, and this can be bad. You won't build the protein, and it could even be fatal for the cell. So the example I have here is UGG. It codes for tryptophan. And if the G is altered and becomes an A, so you have UGA, you'll end up putting a stop codon into the protein, and then you won't build the rest of the protein. So a point mutation is a mutation of a single base pair, so at one point in the gene. So right here, there's a C in one and a T in the other. And this would be an example of a substitution. And substitutions or point mutations could be a silent mutation, they could be a missense mutation, or they could be a nonsense mutation. So all the types of mutations we've talked about so far are examples of substitutions, which are point mutations. Now, frameshift mutations are really bad because it results in the entire reading frame to be altered. So this could be due to a deletion, so you remove a base. So instead of being ACG, TACG, you get ACG, ACG. So you miss an entire base pair. An insertion is similar, so if you put a G right in between C and T, you would change your reading frame. So the codons will be read differently, and that will be a problem. So this is really bad for a protein. Our next type of mutation is an inversion, and in this example, a section of DNA is cut out and flipped over. So in this case, we have ACG, TACG, and this part gets flipped. So instead it reads ACG, TGCA. So again, an inversion is just another form of a mutation that can happen where the code gets read differently. You probably won't put the same amino acid in there because ACG and GCA aren't going to be the same codon for the same amino acid. Translocation is when a piece of DNA breaks off and is added to a new location in the gene or even onto a different chromosome. So in this case, we've chopped off the CA and put it at the end in front. So again, you're going to have a different amino acid being inserted all the way along the way because you've moved an entire section and moved it somewhere else. So I find this is a handy memory tool to keep track of the different types of mutations. So imagine that the word beast is our normal gene. If we substitute an F for a B, you end up with feast. If you insert an R between B and E, you end up with breast. If you delete the A from beast, you get best. And if you invert the last two letters, you end up with beats. So these are all examples of the different types of mutations that can occur.